rid of stats tables. I tried really hard, but this is the best I can do for getting rid of the numbers. But here's the point of this one. It turns out that if you guys remember way back to when you took, 9th grade biology or college biology, you would have learned about a thing called a chi-square test. And what a chi-square test does is it looks at, in a random world, how many you would expect in a certain category. So our categories here are the environments in which these species live. Near shore, inner platform, middle platform, outer platform, open shelf. So what environment were these bracts living in? And then we're going to look at whether they survive or they go extinct. So based on the way that this process works, you can determine how many you would expect to be in each of these categories. And that is the number in yellow here. And then that's the number in this teal color. And then you can also, of course, count how many actually do this in your data set. And that's the number in yellow, that's the observed. Now, you can statistically test whether what you observe and what you expect are different than each other. And what we find here is that species that lived in the middle and outer platform survived much more than we would expect, while species that lived in the nearshore and inner platform go extinct much more than we would expect. So not only is survival through this interval related to how big your range is, but also matters where you lived. If you were living in the deeper water environments, you had A, a bigger range, but B, a bigger chance of getting through because your environments were less affected by these changes. All right, so let's look at the invasion events. Well, these are pretty easy to map. Because we now have our species in a way that we can actually track them through time, we can see where they started, we can see where they moved, and we can determine where we've got invasion events. And so this species, Pseudastraca devoniana, is another really common guy, started here, in kind of the New York region. Later in time, you can see the scale bar here, moving upward. Later in time, moved into Iowa, and then after that, invaded over here into the New Mexico basin. Now, each of these episodes of invasion actually turns out to be tied to one of those transgression or sea level rises that we saw on that sea level curve. So this is providing a mechanism for getting these species around. They've got across those intracratonic basins there, or the arches that we saw, and sea level rise is giving them a chance to do that. Yeah?
The little area where there's just a straight line, is that just because you don't have data there? Would they have been across that whole range? Probably?
Right. Well, that's a good question. The question is, so with the little straight lines, what really is the interpretation there? Well, those are areas where we don't have Devonian outcrop. So we really don't know for sure if they lived there or not. We assume that they had to somehow make that transition, and that at the time it was wet. So we make it with the smallest possible line possible, just to indicate that it was there, but so that it doesn't add very much range lines to the data set. Okay, if we look at these invasion events through time, you can see that there were certainly times that had higher invasion intensity than others. You can see these little pink arrows also correlate to those transgressive events that we saw on the sea level curve. So again, these invasion intensities are related to the fact that species had to cross the barriers. There had to be a mechanism for that. And so the primary mechanism here during the Devonian at least was the ability to have sea level basically swamping and providing a mechanism to get across these intracratonic basins. And so if we look at the relationship between invasive history and survival, it turns out here that the species that have one or more episodes of invasion in their history are preferentially surviving this mass extinction or biodiversity crisis. So again, being an invader is a good thing to do if you want to get across one of these times of ecosystem reorganization. Okay. So that's one way you can assess species ranges, is by actually just drawing these little polygons around there. Now, one thing, of course, that you have immediately recognized, because you're very familiar with the fossil record, is that, well, what if your rocks don't crop out there? Maybe the species live there, but the rocks are in the subsurface or the rocks are eroded. So we'd like to get a little more detailed and have a better predictive ability rather than just drawing some simple polygons. So let's think of more sophisticated ways to do this. And this is the part that we want your help with on the Cincinnati project. So we're going to be dealing with something called a fundamental niche. The fundamental niche is the area in the theoretical ecospace where a species can live. So every species has environmental tolerances. So my hypothetical brachipod has certain salinity. It can't live in high salinity, it can't live in low salinity, it can't live if the temperature gets too warm. And so it lives within this set of variables. Now, realistically, it's a much more complex set of variables. 2 variables never determine anything. It's going to be more complicated than that. But this is the basic idea we're trying to figure out. What types of situations can these different species live within? To do that, we're going to use a fancy computer program called GARP. GARP stands for, as you can read, Genetic Algorithm Using Rule Set Prediction. What this means is that we're going to take data in our data set about where a species lives and what the environment is like across the range, and we're going to model, based on that information, where the species should live. And we're going to do this through an iterative approach using multiple algorithms in a way that, let me just say, is a really good way to go about doing this. Takes in a lot of mathematical uncertainty and does a really nice job. This was originally designed to do with modern fossils, or modern animals. And so, mostly with birds. And so this is a cattle egret, which is a species that's native to Africa, but has been introduced to North America. And so with this particular example, all of these little dots here represent known occurrences in Mexico. And then all of the little X's, and sometimes X's are so overwhelming that they make black boxes, is where, based on the Mexico points and the environment in that region, you would expect the species to be able to live in the United States. And if you look at that and you see how accurate it is in fact insanely accurate. So this p-value means that there is a 1 in 10 to the minus 200, so 10 with then 200 zeros after it, chance that this was a random occurrence. So really high accuracy when we're looking at these types of results. So, of course, like my other stuff, it had never been done in the fossil record before, but why not? We know where species occur, we can use sedimentary information, we can understand environments of deposition, we can do all the same type of information we have in the modern, let's apply it to fossils. So, I took the data from our first case study, and I made it smaller, so now we're really just looking at the Appalachian Basin. So here's the Devonian outcrop belt, as you can see, running through New York, Pennsylvania, down here into Virginia. So all of the little diamonds here are those species occurrence points that we started with last time, collected from museums. And the blue dots represent locations where I have stratigraphic information that I pulled out of stratigraphic columns from things like field guides. Okay, so that's data collection. From all of these little environmental data points, I can interpolate out environmental variables. So from the rocks, I can interpret what's going on with water depth, for example, as one of my variables. And so water depth is, of course, shallow, where it's light-colored, and it's very deep, where it's dark-colored. And this matches really well with what we know about the rocks. So if you've got a very dark shale, that would indicate deeper water than if you're looking at something like a deltaic limestone, or a deltaic sandstone. So using what we know about the environment to work this out. And then from this data, from where a species occurs, plus what's going on with the environment, we can use this program and then model where the species should occur. And so the way to read these maps here on the right is that this dark blue indicates a 0 probability output from the program the species should live there, and this dark orange indicates kind of 100%. And so that the colors in between dark blue and orange are somewhere in between 0 and 100, depending on where that color lies on that range. Okay. So for this study, again, I looked at the Appalachian Basin, and to split up the environmental variables, I put them in half-degree grid boxes. In the modern, they actually do 1-degree grid boxes. So this is actually at a higher level of resolution than a lot of the modern studies. And we looked at 3 time periods. Because we really couldn't do all 19 this time, we had to break it down. So again, we're looking at this guy, which is basically the Penn-Dixie-Corey equivalence, down here in the Devonian, up here before the crisis interval, and then during the crisis interval. So again, same set of brachipod data, but we needed to get get enough data points that were distinctive. And so we ended up modeling 20 species in the late zone, 9 in the middle, and 10 in the early zone. For those of you who really enjoy Devonian stratigraphy, these are the units that we used in these different time slices. So here's the Ludlow. Okay, these are the different